All right, we'll go ahead and get started. So, couple of quick things. So I handed back the grades on the homework assignment. So you'll see on the back side, I put additional notes on there. One of the things that I'm going to try to do is use something that, well, they use in elementary school now, this 1 to 4 scale, okay? So, what is this? Here's the explanation. It's a little small on here, but basically this is not going to be this is not the grade, but this is kind of for you to orient yourself on like how, without grades being included, without the points being included, how are you performing on the individual assignment? So the 1 to 4 scale, 1 basically means like you're not meeting the expectations that I have. A 2 means, yeah, you're somewhat there. Usually this is like errors in terminology, maybe pronunciation in this case of this assignment. Sometimes the information might be incorrect. 3 means you're meeting the expectations. Mostly the terminology is correct. The errors are really minor. They don't affect the overall. They don't affect the grade. And then a 4 is like you're going above and beyond. So like folks who had 4s on this past assignment, or that I felt earned a 4, were ones that were well organized, clear. The terminology was used really, really well. They actually went beyond what the questions were asking and started including things like, there's 3 protomers, therefore the T number equals 3. That's not a connection that I was asking for, but that some people ended up including, okay? And so that's really, really good. Use this kind of as a guide for yourself to kind of get a self-assessment of where you're at. The other thing to point out is use it as motivation, okay? I hate it when people tell me that like I'm only so good, so I want to prove them wrong, okay? So use this as motivation if you want to. All right, so that's the first part. Second thing, I just wanted to point out real quick what the next assignment is and where you can find it. So we're going to do a lit review on Friday, and if you go under content and you go into our viral structure and classification, oh no, that's not the right one, sorry, that's what we just finished up, molecular bio. Lecture 3 here, this will just take you directly to a PDF of the article that we're reading. That's the lit review article, okay? Now, am I expecting you to know everything about this paper coming into class? Hell no, because it's kind of complex, okay? The goal is by the end of class that we understand the majority of the paper, okay? And there are going to probably be things in here that you have never seen before, and that's okay. Goal for us, at least early on here, is to walk through these individual papers and walk through and decide, how do we actually read these? What's the important information to pick out? So to make sure that you're spending time with it ahead of time, the assignment is in here, and it's this critical thinking 1, or no, it's not, it's this lit review, sorry, I'm totally all over the place today. And if you just click on begin, now it's got the questions. And you can type these questions. You can type the answers to these in a Word document if you want to, and just copy and paste them right into here afterwards. So, for example, the first question is: describe ribosome scanning model of cap-dependent translation. What's the role of the EIF4F complex in this process? Guess what? We're talking about that today. Okay, so this in some ways is kind of a review question to make sure that you understand what we've talked about earlier this week when we discussed the paper on Friday. Grace, do you have a question?
Yeah, so once you begin, can you like close out and connect?
I believe that you can. I think you can click on Save All Answers, and it will just save it for you, and then you can close it and come back in and continue to work on it. To actually submit the assignment, you have to hit that red Save and Submit button on the far right-hand side. Okay? Other questions? And I think I have this set up so that like you could go in as many times if you needed to. That's where like if you copy and paste it from a Word document in and you're like, oh shoot, I forgot to include a part, you can just recopy those answers instead of having to retype. So here's our basic mRNA structure. 5' cap, couple of poly A tail on the end. Couple of other things I want to introduce real quick. 5' UTR, which stands for untranslated region. This is the region between the cap and the start codon. 3' UTR, region between the stop codon and the poly A tail. These have regulatory elements in them. Who's heard of microRNAs? A few. MicroRNAs bind to these UTR regions. Coding sequence, also known as the open reading frame, everything between the start and the stop codon. That gives us our protein. The other thing to point out is that there is a sequence upstream of the first start codon known as the Kozak sequence in eukaryotes. Prokaryotes, they have a similar functioning sequence known as the Shine-Dalgarno. Okay. The purpose of the Kozak sequence, which was identified in the 80s, I'm like skipping ahead because I'm trying to find it real quick. I'll go back in a couple of slides or a couple of minutes, but I just wanted to point out this is the Kozak sequence. It's this GCC or GCC. AUG is your start codon, and then there's this last nucleotide afterwards, which typically is a G. And what we mean by consensus is if we look at all of these nucleotide sequences just before open reading frames of that first AUG and just after, this is the most common sequence. Are there variations? Yes. What are the effects of those variations? Well, they can either increase or decrease the likelihood that translation begins at that AUG. We'll talk about the importance of this Kozak sequence in a little bit. But Sabrina, to the conversation we were just having where viruses can have a single mRNA and encode for 2 proteins, you can see here where maybe that might happen by having 2 AUGs but with different Kozak sequences. And one is a stronger or more close to the consensus sequence than the other one is. And so that would promote translation from that one more often than the second one. Jonah.
For the single mRNA that has 2 proteins, would you have like an untranslated region in between the 2?
Not necessarily. Not necessarily. Actually, in eukaryotes, we have what we describe as monocistronic mRNAs. Each one encodes for a single protein. So typically, but that's different in bacteria. Bacteria, you can have a single mRNA that is polycistronic, has separate start and stop codons for each of the proteins being produced. So viruses in eukaryotes have to have some unique features. The different Kozak sequences can be one of those. We'll talk about some other ones as well. Poliovirus is a great example of another one that produces what we call a polyprotein. So we'll take a look at that, I think, on Wednesday. All right. I'm going to go back a few slides. Sorry to skip ahead. Okay. Codon code. Each 3 nucleotides, a codon in the open reading frame encodes for an amino acid. I will never expect you to memorize this, but you should know the 1 start and the 3 stops, right? 1 start, AUG. 3 stops. Remember, those of you who have had me in the past, I love to think of these in terms of colleges. UGA, University of Georgia. UAA, University of Alaska Anchorage. UAG, something in Guadalajara. I cannot remember. Okay? So those are our 3 stops. I just want to pause real quick and see if there are any questions. I know I'm throwing a lot of general information today. And again, kind of put in a context, the reason to do this is because this helps us to understand a lot about every single virus down the road. So I'm going to pause real quick and see if there are any questions. Go ahead, Grace.
I missed kind of what the particulars of the 3' UTR.
So the 3' UTR we can think of as a regulatory element. Oftentimes in the 3' UTR, it contains, this kind of goes beyond what you need to know, Grace, but it contains typically binding sites for microRNAs, which would inhibit translation.
Does that do with the Kozak sequences?
What's that?
Does that do like related to the
You know, it somewhat is. It's different in that you still get the ribosome complex forming with the mRNA. It just stalls the ribosome from proceeding with the process of translation altogether. And microRNAs have really become predominant in eukaryotic genomes, particularly humans. We're looking at them in terms of like disease, etc. Some viruses encode them, but it's only the larger ones. Other questions? Jonah.
So the Kozak sequence can increase or decrease the likelihood that an mRNA is translated?
Yeah, you could probably, I think maybe the best way to say it is, if this is our general consensus one, there can be what we call up mutations or down mutations. Up mutations would be deviations from the sequence that increase the likelihood that translation starts at this AUG. Down mutations would be ones that are different from the consensus sequence that decrease the likelihood this AUG is the first one to be, is the one to be translated from.
Okay.
Yeah, absolutely. Yeah. So this is our general Kozak sequence. We can have up mutations, ones where the mRNA sequence is different and increases the likelihood that translation begins from the AUG, or down mutations, ones that deviate from the consensus sequence and make it less likely that translation will begin from this AUG. And it seems, just a quick side note, it seems the most important ones are this G here and the decision on this A or G seem to be the most important ones in determining the likelihood that translation begins from that AUG. Annie.
Is this only if the two start codons are the same reading frame? Or can this shift the...
You mean start and stop codon, right? Oh, no, sorry. They do not, no, I see what you're saying. So you're asking like if two start codons...
Are out of reading frame?
Yeah, that's a great question. They don't have to be in the same reading frame in the case of the Kozak sequences.
So like, yeah, so is that the affinity for the Kozak sequence that...
Sorry, bad
is
ankles.
that strong enough to shift the reading frame, I guess, for those?
Yes, it is. And actually there's a number of different ways that viruses can shift reading frames as well. HIV is a good one to study for that. We'll try to take some time near the end of the semester to discuss it. Here. Jonah and then Andrew.
Is that AUG like the start codon for the whole mRNA, or is that before?
That would be the start codon for the mRNA. Okay. So anything before that, this Kozak, or at least part of it, is in that 5' untranslated region, or the UTR.
Okay.
Andrew?
So the Kozak sequence just increases or decreases the probability the translation will start?
Yeah.
AUG? So is there times where this process messes up and the translation occurs at another AUG?
Absolutely. And I think that's a great way to describe it, is it increases or decreases the probability. I think that's a really nice way to phrase it. That's perfect. Thanks for bringing that up. Okay. I just want to bring up this idea of reading frames real quick. I think some people know this, but I just figure I'd take the time. So here's a very simple example where we have an AUG start codon, and then we have 3 stop codons, or what could potentially be stop codons downstream. And the easiest way to look at an mRNA is break it up into the codons, chunk it up, and so that's what I've done down here. Taken this sequence, put a couple of spaces in between each, and what you can find then is our open reading frame is where we have both a start and a stop codon in the same reading frame. That's our open reading frame. This UAG here is actually split between 2 codons, so that one would not be recognized as a stop codon. And then this UGA at the end is split up again between two potential codons. So the point of bringing this up is you can't just say, oh, here's the start, here's the stop, that's the length of the protein. You have to break this up into codons first in order to decide where the in-frame start and stop is. This becomes important, especially when we begin to look at bacteriophage genomes later on. Okay? Luckily, we got computers to do this for us, though. Okay. Maybe the last thing to bring up for today then is what is cap-dependent and what is cap-independent translation. And this is where we begin to talk about the EIF4F complex. As the name implies, cap-dependent translation is dependent on the mRNA cap, specifically the 5' cap of mRNAs. That 5' cap, we'll take a look at in a minute, helps to recruit what's known as eukaryotic initiation factors, which can be abbreviated as EIF, lowercase e for eukaryote, IF for initiation factor. The initiation factors help to bring the small and large ribosome subunits to the mRNA to initiate translation. Some viruses will use will have a 5' cap in their mRNAs. Some will actually steal host 5' caps. That's how influenza works. It decapitates host mRNAs and takes that cap and puts it on itself. Really cool. It's called cap snatching. It's a horrible name, right? And then some actually have a completely different mechanism known as cap-independent translation. And that's what this one down here is trying to show you. So what cap-independent translation is a mechanism that doesn't require a 5' cap. It requires something like an iris or an internal ribosome entry site. And we'll talk about this in a few minutes. We'll try to finish up with the iris today. So here's how translation begins. I have it written in words here. I'm going to skip and just go straight to a model because I typically work better from pictures. But if you want the explanation, it's on the previous slide. We have a complex known as EIF4F, which consists of these proteins here, EIF4E. This is known as the cap binding protein. Its function is to bind the 5' cap. EIF4G is a scaffold protein. So it's going to bind together EIF4E, EIF3, PABP, which is the poly-A binding protein, and then EIF4A. Okay, I'll pause there for a minute.
Can you say what that one does?
Which one? The 4G? It's a scaffold. So think of it as like on this one, it's got binding sites for EIF4E, it's got a binding site for 4A, poly-A binding protein, and EIF3. 4A is a helicase. So the role of 4A is to actually kind of serve between the mRNA and the ribosome, a way to unwind any secondary structures that exist in the RNA. So break any bonding that hydrogen bonding that may occur, to make it easier for the ribosome to load onto the mRNA. Poly-A binding protein here, note the mRNA. This is not what you have seen in the past probably, where we have just this like linear mRNA where the tail's hanging off in the end. mRNA adapts this circular formation when in complex, and this seems to help load the ribosome onto the mRNA. It increases the efficiency. It also helps to protect that mRNA. makes it less likely that it's going to be degraded. We'll come back to poly A binding protein when we talk about poliovirus. And then finally, EIF3 here is associated with the small subunit of the ribosome. And so when EIF4G binds to EIF3, that helps to recruit that ribosome to this complex. Okay. So the goal of these initiation factors is to recognize the 5' cap, to scaffold a bunch of proteins, and ultimately recruit the small ribosome subunit to the mRNA. At this point, the ribosome begins to scan, and it just starts to move down the mRNA in a 5' to 3' direction. When it recognizes a Kozak sequence, another set of initiation events occurs, which help to recruit this large subunit, and now translation begins. We're not going to worry so much about those other events right now. Okay. Blocking any of these, and our first paper deals with blocking one of these. Phosphorylation of this guy right here, EIF4E. But blocking of any of these steps could lead to a blocking of cap-dependent translation. Cap-independent, on the other hand, oftentimes can bypass some of the required machinery for cap-dependent translation. In cap-independent, one of the most visible structures is known as an iris, or again, an internal ribosome entry site. These are complex secondary and tertiary structures of RNA. There does not seem to be any sort of pattern as to what these look like. This is the example from hepatitis C virus. And somehow this helps to, this structure right here can bind to the RNA polymerase, or can bind to, for example, the EIF4G to help still recruit the RNA polymerase. The goal of irises are to make translation independent of the cap. And you might start to see where this becomes very beneficial to a virus. A virus could stop cap-dependent translation and yet still encourage for its own translation to take place by encoding one of these irises. We'll come back to that later on. Note here that the EIF4F complex is slightly different. In this iris example, we have EIF4A, G, poly A binding protein, EIF3, and then what's known as an ITAF, and these are just, these are iris, I can't remember what it stands for, but these are a class of proteins that typically seem to be associated with the iris as well in these processes. Okay. I think we'll stop there for today. That's a good point. All right. So lots of information. The things to keep in mind, probably the takeaway points today. Start to think about where we're going with this is how can viruses use these different systems to overcome some of the constraints that are put on it by the host. One of the things that we have not had time to talk about today, and maybe we'll get a little time on Wednesday to at least start to bring it up, RNA viruses, they trigger an antiviral response in the cell. RNA in the cytoplasm, unbound, is not normal in a eukaryotic cell. It's recognized as being foreign. Typically, the host response, shut down translation. Start to think about how can a virus overcome that. And some of those examples fit in even with what we've talked about today. All right? All right, have a great afternoon, everyone. Or a great rest of the day. It's still only 10 o'clock, so I'll see you on Wednesday. Yeah, Amelia.